Today we will start with a very important chapter, that is principles of inheritance and variation. In this chapter, we will learn about the discoveries made by Mendel and also its enhancement. We already know who is Gregor Johann Mendel, right? He is the father of genetics. He has given us the explanation of heredity. Now, what do we mean by heredity? The offsprings always resemble their parents, and this is because of the process of heredity. So what is basically heredity? The tendency of individuals to resemble their progenitors, that is their parents, is called heredity. Heredity includes different characters. These are the characters that are inherited or transmitted from one generation to another, and they are fixed for a particular organism. So, the child always resembles its parents. Next is the term eugenics. Eugenics means a branch of medical engineering. This includes the treatment of genetic disorders. Johansson was a scientist who gave the terms gene, genotype, and phenotype. The characters that provide individuality to a species are known to cause variations among the species. Right? The primary reason for that is that the environment of the organisms is always constantly changing. So in order to adapt to its environment in a better way, living organisms inherit or develop changes. The heredity of an organism interacts with its environment, and this interaction determines what an individual organism is like at a given moment and what it can look like if it develops further in future. Genotype is a term which means the sum total of heredity. Right? The sum total of heredity, including the genetic constitution of the organism that it receives from its parents, is known as the genotype. It includes the observable characters like color, form, size, behavior, chemical composition, structure, and both the external and internal gross and microscopic constituents that form the phenotype of the organism. So what do we mean by genotype? Sum total of genetic constitution. and phenotype is all the observable characters of an organism. Now, when two individuals have the same genotype, that is, their genetic constitution is same, but have different phenotypes, this means that its observable characters are different when they come in contact with different conditions of food, temperature, light, humidity, and other external factors. These occur as a result of environmental variations between the organisms. Sometimes two different genotypes develop because of the same phenotype. These kind of organisms are known as the phenocopy of each other. So what is the phenocopy? It means the organisms that have different genotypes but give rise to same phenotype. Is it clear, everyone? Any doubt? As we can see in the image, the phenotype may be a purple color and its genotype is different. That is, it may be dominant PP, that is the dominant character for purple color. Again, the color will be purple if it is heterozygous, that is, it has one dominant and one recessive gene. But as the dominant character is purple color, the color of the flower will be purple. Right? But the color of the flower will be white if its genotype is homozygous and has both the recessive characters. So the ratio of purple flowers is 3 and that of white is 1. So do we understand? The genotype is the heredity, the genetics, and phenotype is the observable character, that is the color. Now let us understand some differences between genotype and phenotype. Genotype constitutes the genetic makeup of an organism, whereas phenotype are the external morphological characteristics and behavior of an organism. The genotype can be determined by observing the DNA or genotyping methods, and phenotypes are visible naturally. Genotype depends on the gene sequence, whereas phenotype depends on genotype and the environmental factors. Genotype is inherited by the offspring, whereas phenotype is not inherited by the offspring. Genotype consists of all the hereditary information that is expressed and suppressed, whereas phenotype consists of only the expressed genes. Amongst the sexually reproducing organisms, no two individuals have the same heredity, except for identical twins. Only identical twins have same heredity, and so they are known as identical genotypes. Now let us come to some pre-Mendelian ideas about the process of inheritance. Charles Darwin proposed the theory of hypothesis of pangenesis. August Weismann proposed the germ plasm theory. It is also known as the theory of continuity of germ plasm. According to him, living organisms are made up of two materials, the somatoplasm and the germ plasm. Darwin, as we know, proposed the theory of pangenesis. he proposed that gemmules or pangenes are produced inside the tissues of the parent that are further incorporated into the eggs of the females and sperms of the males. So when they fuse, they are transmitted to the offspring. So this is the body of the parent from which the gemmules are released and they enter into the egg. They are then further incorporated inside the developing embryo, and so the hereditary characters of the parents are visible in the offspring. Right? Is it clear, everyone? Somatoplasm makes our entire body. So what is our entire body made up of? According to the hypothesis, it is made up of somatoplasm. It undergoes growth, development, and finally it dies. Then germplasm is responsible for continuing the progeny generation after generation. Mendel also proved that heredity by transmission of units in a reproductive cell occurs. So, the reproductive cells of the parents inherit the trends into the offspring. And he finally put an end to these vague notions about the theory of pangenesis and germplasm theory. In 1900s, three scientists rediscovered the work done by Mendel independently. The first one was Hugo de Rice. He discovered it while working on an evening primrose. The second scientist was Carl Correns, who worked on Z maize and found the work of Mendel. And third was the Chesmark, who worked on different flowering species and discovered Mendel's work independently. Mendel had performed his experiments on garden pea plants. Their scientific name, as we all know, is Pisum sativum. The works of several plant breeders then showed similarity with the work discovered by Mendel. Mendel said that the traits are hidden or masked for one or more generations, and only then they can reappear in the next generation. Okay? In order for the traits to appear in the next generation, they should be masked in one or more generations. The traits may also remain together in one generation and separate in the later generations. One form of a particular trait may be seen more often than its alternate form. So he developed three basic characteristics of inheritance. The first one is the traits being hidden in order to reappear unchanged. The trait may be together in one generation and separate in another. And there may be a particular trait that may be more often than its alternate form. Mendel also selected common garden pea, that is Pisum sativum, for his experiments because the pea plants are having a number of well-defined contrasting characters. They are also bisexual, so the male and the female flowers are located in the same plant. They undergo the process of self-fertilization, and hybridization or crossing over in them is easy. Emasculation can also be done for crossing over. Mendel selected 7 contrasting characters. The first one is the seed form, that may be round or wrinkled. The second is the seed color, that may be yellow or green. The pod form, that may be inflated or restricted. The pod color, that may be green or yellow. Flower position, that may be axial along the stem or terminal on the top. The seed coat color, that may be grayish brown or white, and the length of the stem, that may be tall or short. So, here are the 7 contrasting characters studied by Mendel in detail. Seeds may be smooth or they may be wrinkled. Here the smooth is the dominant and wrinkled is the recessive. The seed form may be yellow, which is dominant, or green, that is recessive. The cotyledons may be inflated or yellow. Dominant is the purple and red colors, and recessive is the white and constricted colors. The flowers may also be green, yellow, and cotyledons may be inflated or terminal. The flowers may also be present along the axis or along the ends, and they may be tall or dwarf. Okay, this is the flower color, purple or white, the flower position, terminal or axial, stem length, short or tall, seed shapes, round or wrinkled, seed color, yellow or green, pod shaped, inflated or constricted, in pod color, that may be yellow or green. These Mendel's differentiating characters, like the dominant and the recessive, are known as the determiners or the traits or the factors or the genes. Bateson also proposed the term allelomorph or allele. These pairs of contrasting characters are known as alleles or allelomorphs. The dominant one is represented by a capital letter and recessive one is represented by a dwarf letter. These capital and small ones, that is dominant and recessive traits, form the pair of alleles. There are genes that control these 7 traits in garden pea plants, and these genes are located only on 4 chromosomes. Now let us understand Mendel's experimental technique. Mendel conducted breeding experiments in 3 stages. The first one is selection of the pure parent plants, followed by the production of first generation of plants by hybridization, followed by the raising of second and the subsequent generations by self-pollination in hybrids. Let us understand the selection of pure parent plants. Mendel noted that the 7 characters and the breeding were passed from the parents to their offspring. By self-pollination, he raised pea plants that had different traits among them. Seeds from the tall plants produced tall plants, and seeds that were yellow in color in turn produced yellow seeds. So, this kind of trait that is inherited from the parent to the offspring is known as a pure trait. Next is the production of first generation of plants by hybridization. Hybridization can be defined as crossing between 2 plants or animal varieties. In Mendel's experiments of hybridization, the pure breeding plants were used. He crossed the pure breeding plants that had contrasting pair of characters. The seeds that were produced by crossing these plants were considered as hybrid plants of the first generation. So it was the first filial generation and declared by the name F1 by Bateson and Saunders in 1902. So the 2 parent generations that had pure characters were crossed and they formed the first filial generation. Right? Third is the self-pollination in hybrids. The F1 generation hybrid plants were self-pollinated and they gave rise to the second filial generation, which is also known as the F2 generation. So it is the second filial generation or the F2 generation which is formed as a result of crossing or self-pollination between the first filial generation. When the first filial generation plants were self-crossed, the second filial generation came into existence. Their flowers were enclosed in bags and the foreign pollens and seeds were allowed north. They were also collected separately and formed the second filial generation. Right? This is about a brief introduction of the chapter Principles of Inheritance and Variations. We will continue with the chapter in the next session. Read well till then. Thank you.